Hello everyone. Hello. You're tasked with completing this primer design worksheet for Bio 350. Your goal is to find out which one of these primers are good for or good forward primers and reverse primers for the following sequences below. Sequence 1, 2, and 3. You're also tasked with finding out which one of the sequences are deletions, insertions, or the full genomic DNA. But first, let's use a Plasmid editor, APE, to find out which one of these primers are the best suited for forward primers and the reverse primers. So in opening APE, I'm just going to open up a new workspace, a new window. And while I open up this new window, I'm going to just take my sequence from each of the sequences individually, and I'm going to paste those sequences into my workspace. And to keep record or good record of it, I'm going to record it as the specific sequence. And save it into my respective folder. I'm going to do the same thing for each sequence, so opening up a new window for each sequence. So that was sequence 1 we just did, then sequence 2. Just copying the sequence here. Now sequences like this could easily be copied from NCBI, but here I provided you the sequence of the RAC1 gene from Arabidopsis thaliana. Now alternatively, in order to find out, for example, which one of these genes have a possible insertion or a deletion, I could copy these sequences and just paste it in NCBI BLAST, and you could see the differences between the genomic DNA and the actual sequence, and you could interpret or tell which gene sequence has the insertion or the deletion. But we're going to do this using APE today. So in this case here, I'm just going to save this. And do the same thing for the third sequence. Just save it here. After my sequences are saved, then I'm going to take the respective primers individually. Say for example, the first primer here, I'm just going to copy this and search for it in each one of my sequences in order to complete the table below, the table that I provided you with. Say for example, this is primer number 1. I'm going to just paste it in here, and we're going to do the work for sequence 1, 2, and 3 to see whether this primer could be used as a forward primer, a reverse primer. We're going to input the TM, or the melting temperature, the length, and the GC, because these characteristics here are good for knowing which primer is actually a suitable sequence to be a primer. So let's look for that sequence from number 1 in sequence 1, the primer sequence from this sequence here. So by just looking for it, by pressing Control F, you could actually paste the sequence into this window here. Now it's important that you click off this, make it not case sensitive, and you click off this also find reverse complement of string. Because if you click onto this, this will also look for the reverse complement inside of your workspace. Right now we're just looking for forward, the forward primer. So just click on next, leave the rest of the defaults as they are, click on next, and you see in this sequence here, there's no sequence for the primer found in sequence 1. So let's go to sequence 2 and do the same thing for sequence 2. If I look for that same forward primer, you could see that forward primer does exist for sequence 2. So now I'm going to close this box, and I'm going to right click now onto where my primer is and go to new feature, so I could highlight, or you could highlight by pressing where the forward color is and highlighting it. I'm going to label all my forward primers green. And I'm going to just label this up here, or change the title to primer number 1. And this would be forward. So if I label all my forward primers green and all my reverse primers red, then I would know exactly whether they're forward or reverse. And just click OK, and you see my primer is highlighted here. Now if I highlight my primer in itself, it gives me here in APE the TM, or the melting temperature. It gives me the GC content, the guanine cytosine percentage, and it also gives me the length. It tells me also where my primer starts and where it ends, and the sequence, this entire sequence, how many base pairs are in this entire sequence here, 2174. So it gives me all the information for not only the sequence but the primer that I just highlighted. So I'll go to my box now, and inside of my box, is it a forward primer? I would say yes, right? And I'll also record the TM. The melting temperature is 55 degrees Celsius. Now remember, the annealing temperature is calculated from the melting temperature, or the melting temperature is calculated from the annealing temperature, right? So we know that in order for us to have a good primer, the melting temperature should be within about 1 degree from each other, right? 1 degree Celsius of each other. Now the melting temperature could be the exact same temperature, right? So the forward primer and the reverse primer could be the exact same temperature, or about 1 degree apart from each other. So it does have a forward primer, and the TM is 55. And let's look for the length. The length here is 20 base pairs. And the GC content, we said GC content was 50. And remember, the characteristics for a good primer, the length would be about between 18 and 25 base pairs, and the GC content would be about 38 to about 50 base pairs with the guanine and cytosine. It would also be good if your primer started with a C or a G in the beginning, and that's for the forward primer in the 5' to 3' direction, because remember, guanine and cytosine has 3 hydrogen bonds that hold them together. So since we have the forward primer for this one here, it is a forward primer. We don't know yet if it's a reverse primer, and we did not check sequence 3 yet. So let's do sequence 2 first. Complete at least sequence 2. Oh, that was actually sequence 2. So that was sequence 2 we just did, sorry. Sequence 1, this was not a forward primer, right? So this was not a forward primer, so we can't put this information in here, because we have no forward primer for the sequence 1. We do have it for sequence 2 here. Let's check or search for this same primer for sequence 3. That primer does not exist for sequence 3. So we will put no here also. No for sequence 3. Now let's look for the reverse complement in each one of our sequences here. Search first for sequence 1. Now we could click on this box here to find the reverse complement. Reverse complement for that primer does not exist in sequence 1. Let's do the same for sequence 2. It does exist here. It's highlighted. It's the same primer, could be used as a reverse complement. So in this case here for sequence 2, we will put reverse. Yes. Now that is not always going to be the case, right? Oh, it's actually in this sequence here, because we had no reverse primer for sequence 1. So we can't put the TM or GC for that sequence. Let's look for sequence 3, whether it's a reverse primer here. And we see it's not a reverse primer there, so we're going to put no here. And take out this information for TM or GC and length. Now you do the same thing for the rest here. And then you select which one of the primers from 1 to 10, is actually going to be the best for forward and the best for reverse, based on the TM, the length, and the GC content, and also whether it appears in all three sequences, because your primer is not going to be used to amplify a specific sequence if it's not existing in that sequence. So the primer has to be there for all three of the sequences, whether it's a insertion, a deletion, or a full genomic DNA. Because if it's not in the deletion, right, we would not know if when we amplify the specific sequence, if there's no primer to amplify that sequence, whether it's a deletion or an insertion or not. So we need to have the primer in all three of the sequences, first of all, and then we need then to check whether that primer is has a TM that is within, for the forward and the reverse, within 1 degree of each other or the same melting temperature, and if the GC content and the length are within the range that we restricted for our domain. Now we do that for all of these sequences here for all of the primers. A good primer, or the correct primer, will have all of the sequences being, whether it's forward, all of the sequences will have forward, and all of the sequences will have reverse for a good reverse primer. So it's either a good forward primer or a good reverse primer for these specific primers that we have here, right? So let's look at some other things about APE. After we complete this table, then you will be able to answer the questions that you had to answer. Let's now go to APE and learn a little bit more about APE. So we have all three of these sequences here. Now, in these sequences, our primers are either going to be labeled for one color, in my case here, green, for all the forward primers, and then it'll be red for all the reverse primers. So I could easily identify all of my primers on all of my sequences to fill out my table. Now, understanding how APE works, there are different, in this tab up here, you could look at these tabs and tell exactly what, or know exactly what to do using these sequences here. One of the things that you could do, even without looking at the tabs, is by looking at the information that you're given within the sequences themselves. Say, for example, you look at these sequences here. These sequences here tell you whether there's an insertion, a deletion, or it's full genomic DNA. How am I going to predict that or assume that is the case? Look here for sequence number. The amount of nucleotides or bases that we actually have here is 2042 for this first sequence. The second sequence, we have 2174. And the third sequence, we have 1684. I could assume by just looking at these numbers that this sequence is the deletion. And that sequence here is the insertion, and that sequence is the full genomic DNA. But in order to identify that fully, I'm going to take one of the sequences or all three of the sequences and go to Tools up here. And then I'm going to align sequences. Now you could align them two at a time, but in my case here, I'm just going to align all three sequences, 1, 2, and 3, sequence 1, 2, and 3, right? I'm just going to align them and look for the selection only, not the reverse complement. And so I'm going to select all of these here, and I'm going to align them to see whether they work or not, or what where they fall, and they have similarities to each other. And after I press OK, I see all of my primers 1, 2, and 3. So one is this primer on top here. The one on top is primer 1. So what you're going to do is you're comparing everything up to primer 1. So this is primer 1 compared to primer 1 here, primer 1 compared to primer 2, and primer 1 compared to primer 3. Right? So it's in this order. So sequence 1 primer design worksheet, it's aligned to sequence 1, sequence 2, and sequence 3. So now, these are all the sequences that match. There's a perfect match between all three so far. So you see the matches in this first one here compared to 1 is 2042, compared to 2 is 2041, and compared to 3 is 1684. there are no mismatches. And the gaps, we have 134 for this sequence here and 358 for that sequence down here. So these here seem to be some alignments that do not match for sequence 2 and 3. Let's see where that is. So far we have all perfect matches, except here there seemed to be a gap, and there is a gap here for sequence 2 here and sequence 3. The gap starts from here, 1000. The gap starts at 1091 for sequence 2, and it ends here at 1223 for sequence 2. So you could actually see where the gap is. This is a big chunk that is missing here between those sequences when compared to the top sequence. Let's go further down. You see another gap, but that gap in this case here is for this sequence. This is sequence 3. And let's go further. So the only two gaps we're seeing, or big gaps we're seeing, is here and here, right? So because we're seeing these two big gaps, oh, and look at sequence 3. This is interesting. Sequence 3, there's a big gap from here, from where the red starts, all the way to down here. So you could assume that this sequence here is where the deletion is, right? And so now, since we know this is where the deletion is, I want you to be able to use your alignments now. You could use alignments to do either finding or fully interpreting what the deletion is, or fully interpreting where the insertion is. I'm not going to give this away fully, right? So let's do another example. Let's just align two sequences here. Say I go, for example, I'm aligning sequence 1 and sequence 2. This is a little bit easier to follow. See here, I'm aligning just sequence 1 and sequence 2 to see the differences or similarities between the two. This is 2042 for this top one here, right? And 2174 for this bottom one. This is sequence 2. There are no mismatches, right? But there's 134 gaps. And let's see, so everything is a perfect alignment except when we come to there. There's one base pair that's missing here. That's not really a problem, an issue. But here is where the interesting thing starts. Sequence 1 and sequence 2, there are differences between sequence 1 and sequence 2, starting here and ending over here, right? So it seems that sequence 2 has X amount of base pairs more than sequence 1 when you compare them. So that should tell you something, whether one of them is an insertion or a deletion. After you compare sequence 1 and sequence 2, I would compare sequence 1 and sequence 3, and then sequence 2 and sequence 3, and tell whether there's an insertion or a deletion between the three of them, and tell me how much the insertion is and how much the deletion is, how many base pairs that is. So now we know how to use the align sequences and align two sequences tools, right? These are the tools that we're going to be using here for this APE assignment. Now you could do other things like look for NCBI BLAST sequences that matches these sequences here. Or you could simply just copy this sequence and paste it in NCBI BLAST and find out what these actual sequences are. Another thing I would require, or I could ask you to do later on, is to take these sequences, not only identify what the full genomic DNA is or what organism they come from, because I already told you that, but I could also ask you, where does the insertion come from? What gene is actually the gene that is inserted, the key gene that I inserted in there, and which part of the gene that I deleted from which organism, and which gene it actually is. So that's another thing. You could identify the sequence that is inserted or the part that is deleted and search for these parts in NCBI BLAST and tell me more about these things or more about the organism or the genes that the genes themselves. So far we learned how to open and save new windows in APE. We learned how to identify the forward and the reverse primers by looking at the length, the TM, the GC, and also how to just search for the primers using this tool here. And then we found out how to align the two sequences by using these tabs here, right? And finding out how to find insertions or how to find deletions. This tool could be used to identify primers also. This will be something that we do probably later on, where you could just highlight this specific sequence, just take the entire sequence and go to tools and go to find primers. And then it just gives you a list, or it gives you the the requirements, so you could actually put the requirements here, the length that you want for your primer to actually be. Say we wanted a primer that is 18 or 20 base pairs long, a TM of 55 and a GC content of about 40, and you just search now in the 5' to 3' direction in this orientation here, and you could actually find all the possible good or key primers in that sequence that you want to use for a specific experiment. In the case here, for our assignment, I gave you 10 primers that you could possibly select from, so you could identify the forward and the respective reverse primers for each one of those sequences, right? So these primers, the forward and the reverse, should work for all the sequences. Remember that. So after you complete this table, right? After you complete this table and you answer the questions, you're done. But just another tool that you could use in the future to find out exactly which primers you could use for an experiment by just being given a sequence. You could paste the sequence into APE, restrict particular domains, and find out or find the primers that could actually work. Now, there are tools that do this part for you, like Sigma Aldrich. They have programs that could actually do this for you. But APE is just another great tool to do things like that. If there are any other questions or additional questions based on this tool, you could ask me by email and I'll be sure to answer you. Good luck.